Okay, let's get started. It's really my greatest pleasure to have Dr. Jiang He today here, giving us a seminar in the Spatial Omics sort of seminar series 3. And so Dr. He is the co-founder of Vizgen, located in Boston, and focus on the commercialization of single molecule based whole transcriptome profiling technology. And Dr. He received his PhD from USTC, University of Science and Technology of China, where I went for undergrad as well. And afterwards, he moved to Boston to be a PhD student with Dr. Xiaowei Zhuang, and received the PhD in 2015. And afterwards, he did a little bit postdoc with Professor Sangeeta Badia at MIT. And I think last year, okay, if you heard about the conversation between us just a minute ago, I think last year, Dr. He decided to take a different journey, which is to work on the commercialization of this exciting single molecule phage-based whole transcriptome profiling technology called MERFISH. And so Dr. He received numerous awards, and I think he's featured on the, I believe, in the Forbes 30 under 30, which is very prestigious, I think, for younger generation scientists. And I think he also was the featured kind of class commencement speaker in 2016 at Harvard University. I think also he has very kind of strong passion to really deliver the impact of science and the technology to our society. I think he authored a book I haven't read, but that's really on my to-read list. I think he authored a book to really increase the awareness of the healthcare sort of need in the resource-limited countries and resource-limited regions in Asia. So you can feel the passion he has in both fundamental science and the translational development and commercialization, as well as the impact on the public health. I'm really delighted to have Dr. He here. And without further ado, and then we really look forward to your talk.
Yeah, really, again, thank you so much for Ron and Ahmet for this wonderful invitation. I'm really truly excited and honored to be able to present and share with everyone what we're doing here at Vizgen. As a brief introduction, as we just discussed a minute ago, so Vizgen is really an emerging biotech company launched last year. So we are commercializing a technology called MERFISH that is originally developed by Dr. Xiaowei Zhuang at Harvard University that really enables in situ single-cell transcriptome profiling. So we finished our Series A fundraising last year with $14 million from Arch Ventures and North Pond. And then we moved to a new location near Fresh Pond area in Cambridge, Massachusetts. So as mentioned, my background also comes from Xiaowei's lab. So I did my PhD in her lab and worked on single molecule imaging and super resolution imaging and application in different biological areas. And then I moved to MIT for postdoc and worked on tissue engineering and cancer diagnostics before coming back to help with the commercialization of this technology. So for today's talk, I hope to first really give you a sense of why spatial transcriptomic profiling is such a powerful technique. And then I'm going to walk you through the technical aspects of our platform technology and how it differs from some other spatial technologies. And finally, I will use a couple of showcase examples to really illustrate how you can apply this technology to address different biological questions and gather biological insights. And in the end, if you have any questions, I would really love to hear from your side and see how we could potentially fit into your research workflow. And actually, Vizgen recently launched an early access program. So if you're interested, please do feel free to contact me. So as many of you know, biological tissues are very complex and made of many different cell types that express genes quite differently. And understanding the gene expression profile within each cell and across the tissue is absolutely critical for better understanding of the entire biological system. So in recent years, sequencing has really transformed our way of understanding biological questions. As tissue behavior is driven by the expression of many genes, the ability to perform this multiplexed measurement oftentimes provide critical insights into that biological system. And in recent years, with the advent of single-cell sequencing technologies, such as single-cell DNA-seq and single-cell RNA-seq, we are able to now interrogate the gene expression profile of individual cells and use this information to really understand the cell composition and cell-cell interaction as well. However, the sequencing-based technologies has a fundamental limitation. That is, in order to perform sequencing, you either have to dissociate the cells or destroy the cells to release the DNA or RNA content. And thus, the critical spatial component, which is critical for the tissue function, is often lost in this sequencing-based approaches. This is something that, in terms of understanding biology-wise, the morphology of the cells and the spatial organization, this spatial context oftentimes is quite essential. So as many biological problems do require this multiplexed and spatially resolved single-cell measurement, we do see a huge demand of technologies that can satisfy these three criteria. And commercially available technologies do offer certain benefits, but many of them fall short on sensitivity, resolution, throughput, or multiplexing power. So here at Vizgen, we are really working to provide a solution that will overcome all these limitations and drive insights in basic research and drug development. So as already alluded, so our platform technology licensed from Harvard University is called MERFISH, which stands for Multiplexed Error Robust Fluorescence in situ hybridization. So this is a RNA imaging-based technique that can locate and quantify RNA species at the same time with subcellular resolution. So probably some of you may know that there are some RNA imaging technologies such as single molecule FISH existed before. However, the limitation of these traditional techniques is that the number of RNA molecules that can be profiled and imaged at the same time is very limited, often less than 5 at the same time. The reason for this low plex is because these traditional methods rely on the use of spectrally distinct dyes to tell and resolve different RNA species. And since we don't really have, say, for example, hundreds of different dyes to label hundreds of different RNA species, so that's why we cannot really have that high multiplexing power. So in contrast, MERFISH technology really enables highly multiplexed RNA imaging capable of resolving from hundreds to tens of thousands different RNAs within cells. So there are 3 key aspects that made this highly multiplexed measurement possible. First is complementary labeling, then sequential imaging, and error-robust barcoding. So as shown by this schematic on this slide, to perform MERFISH, we use complementary labeling strategy to label each targeted RNAs with a type of oligoprobes containing different barcodes. And then each barcode is fluorescently detected in sequential round of imaging, as you're seeing here. For example, some of the RNA species in the first round of imaging, it will have a fluorescent signal of on, and then it reads out as a binary code of 1, and then some are not really light up, then it's a 0. And then we will extinguish the signal and then apply another round of readout probes to read out the second round of imaging signal again, and then some are light up, and then we assign a code of 1, and then some are dark, then it's 0. And by doing this sequential round of imaging, then the optical barcode generated from these multiple rounds of imaging with 1s indicating fluorescence signal and 0s indicating no fluorescence, then we'll help to resolve different RNA species. And then this spatially resolved RNA profiling data then gives a physical picture of the cells or tissue of interest, as shown by the picture on the right. What you're seeing here is that these colored dots indicating different RNA species, and then the gray lines are indicating the boundary of the cells. And of course, you can zoom out to much larger tissue areas to get a global sense of how different genes are expressed on a tissue scale. So MERFISH was originally developed by our scientific co-founder, Dr. Xiaowei Zhuang at Harvard University in 2015. So since the initial publication in Science, it has been validated by a list of publications, either coming from Xiaowei's lab or from other independent labs now as well. and it has already brought transformative discoveries in the field. It was also labeled by Nature magazine as technology to watch, really highlighting the excitement it has already generated in the field. So here is a quick summary of some of Murphy's key features. The first one is that Murphy really has exceptional spatial resolution. We can localize RNA transcripts with subcellular resolution here. So this really allows us to zoom in into individual cells and directly analyze the spatial distribution of the entire transcriptome within the cell. And what this also means is that Murphy has true single cell resolution. Secondly, Murphy has very high throughput. We can profile a square centimeter of tissue in a single run overnight, and oftentimes we can profile hundreds of thousands of cells in a single experiment run here. So this really allows us to zoom out on imaged tissue and directly analyze the molecular and cellular profile of the entire tissue at biologically meaningful length scales. So no sequencing, no follow-up sequencing is really needed for Murphy's measurement. You can get all the information in a single experiment run without additional sequencing cost. Thirdly, Murphy's has very high multiplexing power. We can profile and identify hundreds to near transcriptome numbers of RNAs. At the same time, this technology is also compatible with immunofluorescence. We can image multiple protein markers simultaneously. So this is especially important. Having such a high plex power is very important because if you're dealing with precious samples, it could actually potentially save time and precious sample volumes, and you can gather more biological insights from a single experiment as well. And finally, Murphy has high sensitivity. Since Murphy is based on single molecule fish, which is a gold standard for quantifying RNA copy numbers. So it also maintains that single molecule sensitivity. So for people who work in the single cell field, they can probably appreciate why sensitivity matters. Oftentimes, for example, if you have some not expressed genes to be profiled, oftentimes you may not have too many counts to single cell RNA-seq or so. But since Murphy has a high detection efficiency, it really allows us to detect difficult to characterize and not expressed genes with single molecule sensitivity. And it is really these four features combined that I think really set us apart from some other technologies, because we don't really see other commercially available technologies that can do all of these four. So as a fundamental gene expression profiling tool, Murphy at its basic form just report the location and the quantities of RNA species together with the cell boundary information as you are seeing here. But with such enriched information, it really opens up many application areas. For example, we can use this tool to study the gene regulatory pathways since we are measuring so many genes at the same time with high detection efficiency and single cell resolution. And the stochastic gene expression among these cells can be profiled. And this could help to identify genes that are co-up-regulated or co-down-regulated. Or you can use this to profile gene expression in situ. Or even you leverage this information to classify and discover cell types and cell states. And since the location is recorded, you can also use this to reveal cell organization with native tissue context as well. And if you include marker genes that can report activity, Murphy's then can even be used to profile cellular activity and function here. And finally, since this is an imaging-based technique, so Murphy's can even be used for imaging-based genetic screens such as CRISPR screens. So there are many application areas, and we envision that they will be much more common since this is a really new and evolving technology. So for the sake of time today, I'm only going to focus on the middle four applications. And if you're interested in learning how it could be used to study the gene regulatory pathways or perform CRISPR screens and do these imaging-based phenotypic screens and genetic screens, there are a number of applications coming from Xiaowei's lab. So I highly encourage you to take a look. So then I'm going to go through a couple of examples now to really illustrate how we leverage this tool to get biological insights. And the first example really illustrates how Murphy could be used to profile gene expression in situ across large tissue areas. This is the study published in 2018 in Science, also by the first author is also our scientific co-founder and co-inventor of the technology, Jeff Moffitt, who is now a professor in HMS now. So as we know, to understand the tissue function, it is really critical to understand how different genes are expressed at different anatomic sites and how the gene expression profile may change during disease onset, progression, or after drug treatment. So in this example, Murphy's was used to study the expression of 155 genes in a brain region called hypothalamus. So showing here is a coronal section of the mouse brain with the hypothalamus region indicated in the black box. So this is a region involved in a lot of important functions, such as body temperature control or social behaviors, such as mating behaviors. But how exactly different genes are expressed are not really well understood in this brain region. So shown here on the right is a 2 by 2 millimeter fluorescence image. And what you are seeing is the distribution of only 8 RNA subsets out of this 155 gene measurement. And then below are the single color images. So hopefully by just glancing to these pictures, I hope you can start to appreciate the heterogeneity of gene expression in this brain region. For example, the GAD1 gene, which marks the inhibitory neurons, has very different distribution pattern of as compared to this blue gene, which marks the excitatory neurons. So since Murphy has a really high spatial resolution, you can further zoom in and understand the gene expression pattern at single cell level. So basically, for each individual field of view, the high resolution is preserved across this entire tissue slice. So showing here on the right in this zoomed in single field of view images with now 155 RNAs shown in these different colored dots now, and gray line indicating the boundary of the cells. Hopefully you can also start to really visualize the gene expression profile among individual cells as well. So in short summary, so Murphy here, it was used to create a molecular atlas for the preoptic region of the mouse hypothalamus and started to reveal the heterogeneity of gene expression in this brain region. And the ability to profile gene expression in situ with single cell resolution really sets us apart from many existing spatial technologies because with true single cell resolution, you can start to understand how different cell types are expressing the genes, and this could further be leveraged for cell discovery as well, which is going to be shown in the next slide. And so here, again, with this same gene panel, 155 gene panel, so Murphy was used to profile 500,000 cells in the mouse brain, in the mouse hypothalamus. Then the gene expression profile information among cells was used for cell clustering. And showing here is the T-SNE plot that you will often see in single cell analysis field, basically by projecting this high dimensional gene expression profile in data into two-dimensional plot. And through this, there are about 75 different cell clusters identified in this measurement. And what you are seeing highlighted in different colors are the major cell clusters, such as inhibitory neurons, excitatory neurons, and so on and so forth. So to benchmark Murphy's measurement with sequencing, the cell type identified by Murphy was compared with those found by single-cell RNA-seq. And as shown by this correlation plot here, quantitatively, Murphy agrees very well with single-cell RNA-seq. As you're seeing here, that basically Murphy was able to recapitulate and identify all the major cell types found by single-cell RNA-seq. And with data that is not really shown here, Murphy was even able to discover new cell types not found by single-cell RNA-seq. And this is made possible by two key features of Murphy. First, as mentioned before, Murphy's high sensitivity. So this really allows us to profile not expressed but functionally very important genes that can specify cell types, which is very challenging for single-cell RNA-seq. And secondly, the key factor that made it possible is the high throughput nature of Murphy's measurement. This allows many more cells to be sampled so that rare cell types will be more frequently picked up. So in this study, actually, about 1.1 million cells were analyzed by Murphy. So if you wanted to perform these studies by single-cell RNA-seq, the sheer cost of sequencing for 1.1 million cells sometimes will already make it impossible for most labs to perform such a study, right? So as the location of the cells is actually recorded in Murphy's measurements, so it could further be leveraged to catalog and reveal the cell organization within native tissue context as well. So showing here now are the cross-section images of the mouse hypothalamus, and now with each circle, this colored circle indicating the position of all the major cell classes. For example, from the anterior to the posterior of the brain hypothalamus region. Hopefully you can appreciate that different cell populations have a very different distribution pattern in the brain. And furthermore, Murphy's could be readily used to profile cellular activity and states. This is done by including some of the genes that can report activity. So in this example, early immediate gene called FOS was FOS. C-FOS is included in the measurement. And then this really allows us to identify the single neuronal types. As you are seeing here, the red bars indicate the neuronal clusters that are selectively activated during these individual behaviors, such as parenting behaviors, aggression behaviors, or mating behaviors. So basically, in summary, I hope this example really gives you a sense of how Murphy's could be used for cell atlasing, and this application really has a lot of profound implications for biomarker development, mechanism of action studies during drug development, to name just a few here, and it could potentially provide valuable insights for drug development as well. So then the second showcase example I'm going to go to really illustrate the power of this technique for functional mapping. This is a bioarchive preprint paper coming from Xiaowei's lab earlier this year. The first author is a postdoc called Meng Zhang, and the overall design of the study is very similar to the example I have just walked through. So I'm going to skip some of the technical details and only focus on the biological insights gathered through this study. So here again, Murphy's was used to study the mouse primary cortex region, and a panel of 255 genes are actually selected based on single cell and single nucleus, as well as sequencing data, as well as the domain knowledge. And then the brains are sectioned and stained with the Murphy's probes, and then they are used for Murphy's imaging. And then the gene expression profile was used to really do the cell clustering. And as you're seeing here, this is the sort of the UMAP showing the major cell classes are identified. And overall, there is a total of 95 different neuronal and non-neuronal cell clusters identified. So again, this study was really benchmarked with single cell analysis. And as you're seeing here, there is really excellent correlation with the single cell and single nucleus RNA-seq clusters identified. So very interestingly, through this analysis, a major cell type, basically called the IT cells, intracephalic cells, seems to form in this brain region seems to have a very continuous spectrum of gene expression profile. So as seen by the UMAP plot here, there are about 4 major IT cell subclasses as shown here, the L2/3, L4/5, and L5 and L6 here. And by performing subclustering analysis, these IT cells could be further divided into 19 different cell types, as shown by this UMAP plot here. What you're seeing, these colors are indicating the subcell types, subclusters. And very interestingly, as shown by the subclustering results here, these subclusters do not seem to have really discrete and very well-defined boundaries. Instead, they seem to have a gradual change of gene expression profile. So in other words, clustering based on gene expression profile cannot clearly separate these adjacent subclusters. Since the location of all these cells are recorded in the measurement, so one can actually map out the physical location of each subcluster in the cortical region. And by doing this analysis, a very interesting pattern actually emerged from this study. So it was found that, as shown by the picture here, this is sort of the physical picture of how these 19 different subclusters are residing in the cortical region. So basically, these cell types also seems to form a spatial gradient along the cortical depth positions as well. For example, the L2/3 seems to reside on top, and then as we move deeper into the cortical regions, then it gradually gets to L6. And this is highly correlated with the gene expression profile. So basically, this biological finding really speaks for the power for spatial measurement, because only with spatial mapping, you will be able to really understand how similar cell subtypes are spatially organized in complex tissues like in brain. So furthermore, in this study, another powerful application demonstrated is projection mapping for molecularly defined cell types. So in brain, neuronal circuits underlie all critical brain function. And a central challenge in neuroscience is actually to understand how the neurons are wired and how different brain regions are connected with each other. So to understand where each neuron in the primary motor cortex is projecting to, so in this study, a technique called retrograde tracing was used to trace neuronal projections. And as shown by this diagram here, basically 3 brain regions called the secondary motor regions, the primary somatosensory regions, and temporal association regions, TEAs. These are the 3 known regions that will receive input from the primary motor cortex, and then they are rejected with fluorescently tagged chlorotoxin. And then as the chlorotoxin travel back to the primary motor cortex region, then a rhodopfluorescence image was then performed to understand how different cells are uptaking these toxins. And then this was followed by Murphy's imaging to identify and classify cell types. So in the figure showing here on the right, what you're seeing here, the different colors indicates the different chlorotoxins coming from these 3 brain regions, the MOs, SSPs, and TEAs. For example, this is a neuron that actually uptakes some chlorotoxin from the MOs regions. And then on the right, these are the cell types that are identified. The boundary line indicate the cell boundaries. Sorry, the cell types identified through Murphy's analysis. So as shown by the image in the bottom, actually more than 90% of the cells that receive the toxins are the major IT cell types. And then there are some other inhibitory neurons, such as provocamin neurons highlighted here. And as you can see from here, so basically these are the neuronal types that receive this input from these 3 regions. And on the left, you can clearly see how different IT neurons are projecting to different regions. And in short, through this connection and projection mapping, this study was able to reveal a many-to-many neuronal projection for primary motor neurons with other cortical regions. So there are a lot of really other cool results reported in this study. And really due to the limited time, I want to be able to go through all of them in detail. So if you're interested, again, encourage you to just take a look at the preprint paper. And hopefully in short summary, this example also sort of gives you a sense of how Murphy's could be used for functional mapping and study intercellular interactions. So the last example that I wanted to walk through illustrate how Murphy's high spatial resolution allows us to directly analyze the spatial distribution of RNAs in cells. And this is a study published last year in PNAS. So in any cell, due to the post-transcriptional regulations, the RNA species are not uniformly distributed. And this internal organization of transcriptome underlies many essential biological functions, such as cell motility or migration, or synapse formation, or neuronal generation. For example, in tumor metastasis, you may imagine that the leading edge of the cells may contain RNAs differently than the rest of the cell body. And to really understand the cell function, sometimes a better understanding of this intracellular distribution will help illuminate how these cells function. So in this example, Murphy's was used to profile up to 10,000 genes in cultured cells. Shown here is the projection of these 10,050 genes in cultured cells. On the right, what you're seeing is the correlation with RNA-seq. So basically, for Murphy's measurement, we always benchmark with RNA sequencing data if such data is available. And hopefully you can really appreciate the high performance of Murphy's measurements when we benchmark either with bulk RNA-seq or single-cell RNA-seq. So by simultaneously imaging some of the intracellular or glial markers such as ER as well as nucleus, Murphy's in this example was also able to identify hundreds of protein coding genes, intron retained genes, or 10 link RNAs enriched in the nucleus. As you are seeing here, now these fluorescent dots indicate the genes enriched in the nucleus area, as well as all the ER enriched genes. So basically here, this is really a demonstration of showing a really high multiplexing power of Murphy's with high accuracy and sensitivity, and it also shows how we can do simultaneous imaging with protein markers as well. And furthermore, one could actually leverage this intracellular distribution information and use this to specify cell states. As you can imagine that in any cell, the rate of transcription and the rate of nuclear export, as well as the RNA degradation, varies very differently, and then they correlate with the cell state. So since Murphy has the spatial resolution, you can sort of analyze the balance between the nucleus versus cytoplasmic RNA counts. And by doing this analysis, so-called RNA velocity plot, as shown by here on the right, Murphy was able to really was able to use to really determine the pseudo-time ordering of cells as they're going through different cell cycles. For example, going from G1 to metaphase, or so as it cycles through this cell cycle. And actually, thousands of genes with putative cell cycle dependent gene expression was also identified in this study. So I hope these three examples, though a bit short, I hope already gave you a sense of how it could actually be applied to gather biological insights. And we believe this fundamental gene expression profiling tool can open up many application areas. And we are truly excited by the broad potential of its application. So here at Vizgen, our goal is really to enable more researchers to have access to this great tool and help accelerate their research. So since the launch of our company, we have developed a variety of protocols to process samples in different research fields. We hope you can actually leverage some of our expertise if you're interested to accelerate your research and then ultimately gather new insights in your biological system. So in the next couple of slides, I'm going to walk through some of the other new developments that has been performed in Vizgen, and also share with you what does it look like to work with an instrument like Murphy scope. So since Murphy is a RNA image fish-based technique, so the overall procedure to perform Murphy measurement is very similar to a traditional RNA fish measurement, and could be very easy for any lab to adopt. Here, usually to perform Murphy, first you need to select a panel of genes. This usually comes from a couple of resources. For example, the single cell or single nucleus RNA sequencing information, or bulk RNA sequencing information, or your own domain expertise. And based on this information, then you will be able to select your genes of interest. And then the samples that we can process can either be cells or tissue blocks. For cells, you can directly fix it. For tissues, we can slice it onto the cover slip and then do the sample processing. And then the samples are then permeabilized and processed with the Murphy probes, and then it will be put on the microscope and begin the Murphy data acquisition. So all the data are actually, the acquisition is automatic. And then once it's done, there will be also automated decoding analysis. And then by doing this analysis, then you will be able to get the positions of all the RNA transcripts and cell boundaries all together. And then starting from here, then you can start to leverage the existing information and perform some other additional analysis. For example, the cell type clustering that I have just walked through in those showcase examples, or some other spatial analysis such as the gene expression profile. Or if you're interested in performing some spatial statistics, you can also leverage some of the existing tools in the academic research field and basically perform all those measurements. And at the same time, Vizgen is also developing data visualization tools to enable some of those analysis so that it will be very user-friendly for you to use. And overall, I wanted to highlight that in order to really help you with help researchers to do this kind of measurement, Vizgen also developed optimized reagents to enable critical reagents for sample prep and imaging as well. As highlighted before, this is a measurement that has really high throughput. We often can profile a square centimeter of tissue, often more than 100,000 cells per run, and oftentimes, sometimes it can even get to 500,000 cells in a single experiment run overnight. So to enable more researchers to readily perform Murphy, here we have developed a variety of protocols and analysis pipeline to process samples in different research fields, as shown by on this slide. So since the launch of the company, Vizgen has proven Murphy now actually more than 15 different mouse and human tissue types. This include, for example, in all the major tissue types that you can think of. For example, the mouse brain, liver, lung, intestine, stomach, spleen, and so on and so forth. For the humans, we have also developed protocols for brain, colon cancer, liver, lymph node, and our goal is really to be able to enable Murphy's measurements in any cell, in any tissue type. For sample types, we have demonstrated Murphy's in adherent cell lines as well as suspended cell lines, as well as tissue blocks include the fresh frozen, fixed tissue, or FFPE tissue blocks. For example, on the image shown on the left here, what you're seeing is actually a Murphy's was used to profile a panel of 128 genes in mouse FFPE tissue slices. And only 8 RNA subsets are shown here because it's just sometimes very difficult to show hundreds of RNAs altogether. So we only showed 8 RNA subsets here. By comparing the performance of Murphy's on FFPE samples versus fresh frozen sample, we get really high correlation. The correlation coefficient is 0.94 here. Basically, really highlights the high performance of Murphy's measurements in different sample types. And beyond these spatial gene expression profiling measurements, Murphy's analysis has already started to generate many interesting biological insights in different tissue types. For example, in the liver case that I just walked through here, so we were able to map genes that can specify different cell types, such as satellite cells or immune cells. And one gene, which is highlighted in blue color here, this is a gene known to have a zonated expression pattern. Zonation means that, so in the liver, some part of the hepatocytes, they're closer to the central vein and some are closer to the periportal vein. And then because of the nutrient gradient and the oxygen level in the liver, then some genes may have sort of patterns that they tend to have a high expression closer to some regions. So this is a gene that has demonstrated that has a zonated expression pattern. And hopefully you can see from the Murphy's measurements here that this zonation pattern is very nicely captured by Murphy's data. And similarly, we have seen many of these biological insights for other tissues measured, such as the ovary, you are seeing here, bladder, the whole brain mapping, and the human colon cancer mapping as well. And finally, I wanted to mention that although Murphy's was developed as a spatial profiling technique, it actually can also be used to study suspended cells as well. And we have demonstrated Murphy's in mouse and human blood cells, such as PDMCs. So here, the main advantage of Murphy's is really the high throughput and high sensitivity here. We can measure up to sometimes 500,000 cells in a single overnight experiment run and use these cells to classify and identify different cell types that you can find in the blood. And if you wanted to profile, for example, 1 million blood cells using single-cell RNA-seq, the sequencing cost will be extremely high. And oftentimes, due to the low detection efficiency, many node expressed genes don't even have too many counts by single-cell RNA-seq. While in contrast, Murphy's high detection efficiency really enables you to capture many low expressed genes. This really speaks for a lot of functionally important transcripts such as cytokines, which are very important to understand how different immune cells in the blood actually can function, right? So we imagine that this very cost-effective and highly sensitive Murphy's measurement in suspended cells can and will likely have powerful applications in areas such as liquid biopsy in the future as well. So to enable researchers to have access to this powerful tool, so here at Vizgen, we are really aiming to bring an all-in-one integrated solution. So we are developing an instrument to perform Murphy's and associated reagents to enable critical steps of sample prep and imaging. At the same time, we're also offering a service program to our early partners and help process the samples. So the workflow for our Murphy's solution is fairly straightforward. Basically, for researchers who are interested in performing Murphy's, then they design a custom panel of genes and order these reagents, and then they will prepare the samples and perform the automated Murphy's imaging with the Vizgen instrument. And then you will be able to sort of do the automatic decoding analysis and then do the data visualization as well. And hopefully from there, then you can gather biological insights from these samples. And recently, we have launched an early access program, and we are actually already enabling merfish measurements among early adopters already. So here is what our early adopter program looks like. This really includes an alpha instrument capable of reliably performing merfish measurements. Also we provide key reagents critical for merfish, such as functionalized cover slips, merfish libraries that can profile up to 1,000 genes, and merfish imaging kit and optimization kit. If this is a new tissue that you're working on, we also provide that as well. And then Vizgen also provides training and support so that you can really gather high quality measurements through this instrument. So basically, if you're interested, you can reach out to this email listed here. And finally, this is an overview of our team. As mentioned, Xiaowei and Jeff are the scientific co-inventors of the technology. So Xiaowei is also very well known for inventing a super resolution imaging technique called STORM, which won her the Breakthrough Prize. So David Walt is a professor in HMS and co-founder of Illuminate. So our investors are here, and this is where we are. We are at West Cambridge area, so not very close to Harvard University. So really, in summary, I hope this presentation gives you a good overview of what merfish technology looks like and what is capable of doing, and also gives you a sense of what we are doing here at Vizgen as well. So we're really excited about the potential of our technology, and would love to hear how we could potentially, if any of you are maybe interested in adopting such technologies, we would really love to hear some feedback as well. And again, for Rong and Ahmet, I really thank you for setting up this meeting, and I can take any questions from here now.
Thank you, Jiang, for a great talk. I think whoever has question, I believe you are able to unmute yourself and ask. I think otherwise you can also use the chat box to ask your questions.
Maybe a quick question from here. So how would you envision the industry and academic partnership with merfish?
We are sort of, so for a lot of academic institutions, we are hoping to provide an instrument and reagent solution for them, because we believe this is a model that for us easy to scale up. So we are, for now, for the service, we're mainly providing this to pharmaceutical companies for now.
So the researchers, they cannot necessarily get it profiled, right? They have to be pharma.
Can you say that again?
Let's say if you have a sample from humans, can you send it to you and then get it profiled for like 100 genes?
So yeah, this is the service model that for our selected early partners for service program. This is something that indeed. So basically, those who entered into the service program, they will be able to submit the sample and then select the genes, and then Vizgen will be able to perform the service for them, and then we will send back the data report.
I see.
But actually, since we are so at the early stage, we don't really have a huge service program established yet. So we are offering this to many for pharmaceutical companies at the moment.
Mm-hmm. Actually, for this multiplex fish, one of the challenges is the initial library prep, right? Mm-hmm. Unlike RNA sequencing, it's actually very costly, right? If you were to design an experiment, you're looking at a really high cost, right? Like 100 genes even. So actually, and another model would be that you could actually help prepare libraries at a decent cost, right?
So for reagents, this speaks for the reagent that need to sort of perform merfish. You need to have a set of oligopools targeting different RNA species. So the finalized pricing for these reagents are not really finalized within Vizgen yet, because we are really aiming to bring this commercial solution hopefully next year. So we don't really have a finalized price for that yet. But we envision the price range will be very comparable and even more competitive than some of those existing library cost for sequencing, for example. That's the range that we are aiming for. But again, this is a really early stage, so to be determined, I think is probably a way to think about, but it will be within that range.
Yes.
Hi, John. I think I probably want to kind of further expand the first question Ahmet asked, which is the service you can provide to pharmaceutical company, but for academia, whether or not you can also set up kind of academic collaboration program in addition to service program. I think that would be terrific news for many audience here today.
Yeah, so this is actually something, the early adopter program that we're setting up. So we are actually already setting up and enabling some of the early adopters in academia already. So there are already some that are adopting Murphy solutions now in the lab.
But not instrument, but actually.
Yeah, so this is, yeah, indeed, the service-wise, we are starting to explore that option as well as we are scaling up and growing our company. So this is indeed something that we are very keen to set up hopefully soon as well.
Jiang, this is Steven. Nice talk and nice seeing you again.
Yeah, good to see you.
Great. The new capacity you have demonstrated is really impressive. Even though we have our home-built Murphy system, I'm actually interested in your early adoption program. One thing I have in mind is for this program, are you selling the alpha instrument to the early adopters, or if so, are you at the liberty to maybe share what's the cost? I mean, basically, how much is it?
Yes. Well, so basically, for the early adopter programs, our thinking is that we are not really aiming for making too much profit out of it. So the overall cost for the instrument will be sort of the cost of, if you build it on your own-ish, the cost. It will be almost similar, or maybe slightly higher, but it will be very, very similar if you just build by yourself. Again, since we are really in the early stage, really hoping to enable more researchers to have access to this tool. So we are sort of received quite a bit of requests. So in the end, which groups that we will go for, it really depends on the capacity that we have in-house. So we will go from there. In the beginning, we have an alpha instrument. So the early adopter program will be more of our current sort of beta version now. So it will be a step closer to the commercial equipment. So a bit different from the ones that you will probably see from Xiaowei's lab. But overall performance-wise, it will be very similar.
Great, thank you.
Yeah, could you comment on a little bit more biology, where I'm curious about to what extent your RNA profile is consistent with the functional macromolecules, that is the proteins. So I'm curious if you ever done any immunocytochemistry or other ways to look at the proteins of the transcripts you detected, so that we could say, oh, these are not RNAs to be degraded, but they actually resulted in functional molecules in the cell.
Yeah, so yeah, really thanks for this great question. So yeah, indeed, so basically RNA and protein, they are sort of different biomolecules in the cell, and so we haven't done a systematic characterization, but based on some of the literature that we have now, for example, almost 90% of them will actually, the expression profile RNA will match up, perhaps even more than 90% will match up with the protein profile. So within Vizgen, we have done some preliminary analysis. For example, in the tumor case, sometimes you may want to mark tumor cells using immunofluorescence staining, such as like Ki67 staining, things like that. And we did for selected protein markers that we will do some of the imaging at the same time. But we have not really done such a systematic, for example, characterizing hundreds of those, we haven't done that yet. But we believe since the RNA profile, the majority of them, more than 90% of the expression profile at the RNA level probably will match up with the protein. So we believe this RNA profiling will already enable a lot of, for example, to gather biological insights already.
Got it. Thank you.
Hi, great talk. Maybe I have one question here, if I may.
Sure, yeah.
Great. So is there any limitation in terms of which genes you can use, or I mean, are there any, is it like all or some caveats there?
Yeah, so this is also a great question. So since Murphy's is a technique based on RNA imaging, and we're tying a number of oligos on each gene, and that has a limitation in terms of the length. So usually, so in the papers that has been published, so basically you need to have a minimal length of RNA transcripts around 1,000 nucleotide base pairs each. However, with amplification, basically this is already demonstrated in Shelby's lab. I think in a 2018 paper with branched DNA amplification, you can use fewer probes and then target transcripts that are around 500 nucleotides each. So another consideration for how to pick genes is basically that we often encourage to avoid highly abundant genes, just because this is imaging-based technique. If some of those highly expressed genes are there, it may create a molecular crowding. And although there are custom solutions available for this, basically you include it in other sequential rounds. But overall, if you wanted to get it in Murphy's rounds, we would encourage to pick genes that are not so highly expressed.
Right,
For
thank
example,
you.
housekeeping genes like actin that sometimes have FPKM of thousands, and it doesn't really carry too much information of specifying cell type or cell states. We usually don't, we can measure them, but we don't usually include them because it doesn't really generate too much insights there. But if needed, we can include in other rounds of imaging without any problem.
Right, thanks. And maybe just one more question, if I may. So you said that you can process this centimeter square sample overnight. So is this like one sample, or I can do multiple samples there then?
So the instrument takes one cover slip at a time, but on a single cover slip, since there is a really huge imaging area, so you can put multiple tissue slices on a single cover slip. For example, like brain, we can put 2 or 3 whole brain slices on a single cover slip, often 2 without any problem. 3 sometimes, when the tissue slicing was not really done properly, maybe it gets too close to the edge, but then 2, without any problem, we will be able to fit 2 entire mouse brain section onto the cover slips. And for some human samples, sometimes like biopsy samples from human can be very big, right? And fitting 1 by 1 square centimeter, that's doable. But again, the instrument can only take 1 cover slip at a time. So
right.
So that really is sort of the current throughput.
Perfect. Thanks.
Yeah, so I have a question about the science or neuroscience from your data. So I think the Human Cell Atlas, the kind of put together a group of people to just do computational identification of the cell boundary. It turns out that's not trivial. So when I look at your data, seems like in a cell boundary nicely identified, but I don't know much about kind of brain and the neuroscience, but my imagination or impression I had in my mind for a while is the extremely crowded and the cells really kind of contact each other. But from your cell boundary delineation, it's like a tiny round circles quite distant from each other. Yeah, so.
Yeah, this is actually a great question. So for brain, indeed, as you are seeing from the picture here, the cells are actually oftentimes very well separated from each other. So cell segmentation in brain, either in mouse or human, is actually fairly easy. But for some other tissues that have the cells more densely packed, so Vizgen actually already developed a very robust cell boundary staining reagent kit. So this is sort of an add-on reagent to really enable cell boundary staining. And so far, based on the performance in the tissues that we have demonstrated, the cell boundary staining very nicely segments the cells. And there may be some tissues that are extremely dense, that even using DAPI, you cannot really separate those cells. Basically, they're so densely packed. Then often for some of those, we sort of have to rely on sort of just a nucleus segmentation. But most of the tissue with our cell boundary staining kit, it is already good to go. Yeah, so this is something, because as we are really expanding the capacity and the capability of Murphy's, and move into different tissue types, indeed, if we truly wanted to provide single cell resolution, cell segmentation, and having reagents being able to segment the cells is a critical component, and Vizgen has sort of now developed a solution for that.
Yeah, also neurons have like a dendrites and axons. Those often kind of entangled in as a complex network. Are you able to visualize those as well? I know those regions, the MRI expression level are much lower, but actually super interesting if you can visualize the MRI in like different regions of the axon dendrites.
Yeah, so this is also a great question. So yeah, indeed, in this brain imaging here, there are a lot of RNA molecules outside of the cell, right? So this probably come from the axons of dendrites. So with our cell boundary staining kit, we are actually able to see the axons and dendrites. However, segmenting those axons and dendrites in neurons, since they're so closely attached to each other, it's still a bit challenging. For segmenting in brain samples, we mostly rely on the sort of the identifying the soma boundary instead of getting all the projections identified, because oftentimes we don't even have the ability to sort of section and make sure that all the projections are within the 10 micron thickness, right? Because neurons do project really far away in other brain regions. So yeah, we sort of, our reagents can sort of light up that projection, but it's currently not really being used for segmenting axons or dendrites.
Maybe just one more quick question, if I may. So you showed data on the FFPE. So is that limited to like 8 genes, or you can do like also 100 genes that you showed before?
Oh yeah, sorry, I didn't make it clear. So this is actually experiment of more than 100. This is 128 genes to be exact. The reason that we're only showing 8 RNA subsets is because sometimes projecting hundreds of genes is very hard for us to see on a PowerPoint slide. So only 8 with a biological meanings is shown on this slide. For example, some of the genes that marked endothelial cells, immune cells are actually shown in this slide. So in FFPE, we can totally profile hundreds of genes without any problem.
Super, thank you.
But for FFP, would you require amplification, or would that work right away?
We don't really require amplification, but the thing to keep in mind for FFPE is that very similar to RNA sequencing. For all our samples, when we process this, we do benchmark and check the RNA quality. We wanted to make sure that indeed there is RNA over the, for example, by doing some standard RIM measurement or DV200 measurement to evaluate the RNA integrity in these samples. And oftentimes we found that Murphy's performance outperforms the sequencing, because, for example, in sequencing field, oftentimes we need to submit samples with a RIM value more than 6 or DV200 more than, say, 70% or so. Murphy's high detection efficiency actually makes it possible to process samples much lower RNA integrity numbers also. And this is something that we are truly excited, because it seems that FFPE, as long as you make sure that there are RNAs inside and has reasonable quality, we will be able to process it.
And do you think like any retrieval or clearing or digestion protocols are helpful?
So the details of the protocol I cannot really reveal, but overall, so the overall procedure will be sort of similar to some of the existing publications that has come from Shelby's lab. But for FFPE, to process FFPE, indeed, there are some new assays that has been developed within Vizgen that made it possible.
And how about 3D? What's your limit now?
So for now, since we are doing a sectioning on the tissue, in order to do 3D, we are still relying on the physical sectioning, but deep tissue imaging and potentially getting a thicker tissue is something that Vizgen is very interested in developing something along those lines. But I cannot really reveal too much details since we haven't really sort of robustly demonstrated that yet.
Cool, thanks.
Would your beta instrument be a confocal or an epifluorescence microscope?
So this is epifluorescence microscope for now, yeah.
Jiang, I'm guessing the 1 centimeter square per run kind of throughput, that's not a deep tissue, that's just a single plane of imaging, right?
That's actually a Z-scan. So we do 11
wow.
slides.
Oh, wow, okay, great.
Yeah, for if we're just doing a single slice, a single Z focal plan, we can probably image double that or even triple that, but it's limited by how big of surface area we have on the cover strip stem. But for now, we do, since most of the tissue, in order to really capture as many RNAs as possible, we do prefer to have more Z focal plans captured through imaging. So,
Yeah.
We routinely run those analyses with multiple Z positions.
Great, and I imagine you achieved this large area through improving the instruments, right? On the hardware side.
Yeah. Well, the many things to be combined, because throughput actually, if you think of the time it takes, it actually comes from two things, right? The first is sort of how much time it takes for you to finish a single round of imaging, and then the second is how long does it take for you to reset the imaging. So we basically shortened both times for acquiring the images, or also the reset time between rounds.
Great, thank you.
And then maybe a quick final question. Consider other than single molecule, like low resolution imaging to improve your throughput?
Oh, sorry, I missed the question.
Like you currently use, I'm assuming 60x or 100x, right?
Mm-hmm.
But if you were to use 20x, 10x with a decent signal, you would still get some mapping. Barcoding, of course, questionable, but would this be a model or this is out of the scope for you?
So I cannot really reveal too much of the product development on the instrument part, but this is indeed something that we are actually thinking of developing within Vizgen. Basically, currently it's still sort of high resolution, and resolution-wise, that's why we're really having such a high spatial resolution with this high NA objective. But with some of the other assay development or on the instrument development part, we probably can potentially use some lower NA and lower magnification objectives to further increase the throughput.
Sure.
All right. I think thank you very much, Jiang, for the terrific talk. So before we close, I don't think I have to share the screen of flyer again, but Ahmet, you can kind of remind us one more time, so the exciting talks lined up for the next couple of weeks.
Sure.
Okay,
So
thank you again for the invitation. And
yeah, thank you, Jiang.
thanks for this great talk, Dr. He. So basically, we are completing our third talk of the series, and we have 7 more to go. And the next one is Dr. Oliver Bach from Ecoe Biosciences talking about codex. And next we have academia speaker from EMBL Europe, Theodor Alexandrov, for spatial metabolomics. Then we have Bernd Bodenmiller from University of Zurich talking about IMC, the imaging mass cytometry, and also that's now commercialized as Fluidigm machine. Then next we have Dr. Samuel Inverso from ReadCore. I think I see him here today. And then next we have Dr. Joseph Beecham from NanoString. I think I see also Daya Kim from NanoString today earlier. Hi guys. And then after that we have Dr. Wesley Legand from UNC talking about imaging from academia, lattice light sheet. And he's trained by Dr. Eric Betzek, so it will be really fun to learn. Then last 2 speakers will be Dr. Mike Angelo from Stanford talking about MIDI. Also, they have another spin-out called IonPath. Then finally, we have Dr. Michael Schnall-Plevin from 10x Genomics talking about their spatial transcriptomics platform. And we look forward to seeing you guys.
Yeah, thank you very much. Thank you, everyone, for participating in the seminar today, and look forward to seeing you next week.
thank you.
Thank you. Take care. Have a good weekend.
Thank you.